navigate the technical issues. Anyway, I just want to say welcome, everyone. I also want to let you know that we are going to record this. So if you, although I really would love to see your faces, you know, I understand if you want to keep your camera off, and we will stop recording after the presentations and when we get to the more of the meet and greet part. So for now, if you want to keep your camera off, I understand. But I want to let you know about our goal was to have, you know, some people come today, and we have over 50, I want to say 53 people just keep registering and coming. So that is, I'm just, my jaws dropped. Just, I couldn't believe how many people are coming. So I'm really excited to have everyone here today. And I just want to, there's so much we as an organization want to share with you, but in the essence of time, we are going to keep a lot of that information that I would love to tell you today at a minimum, we'll share later. And I want to just move on to our stars of the show, which are Heather and Gemma, who have joined us today. But before I get to that, I just want to introduce myself and the other board members who are on the call with us today. Again, my name is Marnie Warren. I'm coming to you from Denver, Colorado. And I have a 16-year-old daughter with CHD2. And I will turn it over to Brian, let him introduce himself real quick.
Yeah, hi, my name is Brian Broadbent. I'm from Dallas, Texas. I am a co-president of the CCC along with Marnie. And my daughter actually has a deletion of the gene right next to CHD2 called Chaser, and she actually has too much CHD2. And I will hand it over to Stephanie.
Hello, everyone. Nice to see everybody here. So I've got a 7-year-old daughter with CHD2, and I just wanted to say a special thank you to Catherine, who's been doing a lot of promoting of this webinar on our UK WhatsApp group. So thank you very much. I'm not sure if anybody else is on the call.
I think that's it for the board members here this afternoon. I think a lot of people have jobs and working in the middle of the day. I also want to make sure I introduce Laura Marks. She's our new executive director. She just came on in December. And I just want to do a quick, briefly, just let you know that we brought Laura on after our family conference in June. You know, it was such a success, and we were came out of it so excited, and we knew that we had something special going, and we wanted to keep moving forward and take it to the next level. So we really talked to a bunch of other patient advocacy groups, PAGs, and one of the themes that came out of a lot of them was that they all had executive directors, and they all wish they had hired one earlier. And I will tell you, having Laura on board in the last 2 months has already been noticeable difference. You've seen our growth in our social media presence, and just on our map, which is which we consider our registry, or our kind of our how we track our patients, that has grown by almost 50 people in just 2 months. So it's just amazing that when we really start moving, you know, getting the right people in place, then we can do amazing things. So I'm just excited to have Laura on board. I'll let Laura talk a little bit more about herself, and then we'll hit the real meat of the show today. Go ahead, Laura.
Thanks, Marnie. I'm going to speak really quickly. Thanks so much. I'm delighted to see everybody here. I've been here since November, actually, and I'm delighted to see everybody getting involved. And I'm going to talk to you all a little bit after the talks about how you can stay involved and things that hopefully you can help us with and we can help you with, so we can really tackle this together. I am not a CHD2 mom, but I do have a child with a different rare epilepsy. So I am very aware of how the life of a parent in the rare epilepsy world. And with that, I'm going to turn it back to Marnie.
All right, thanks. All right, with that, I'll just introduce, we have Heather and Gemma. There are two leading experts on CHD2 that discovered the gene 10 years ago, or a little over 10 years ago. We're thrilled to have them here today. I know many of you are familiar with their names, but I will turn it over to them and let them tell their story and do their presentation today. And I will also say, if you have questions, please just put them in the chat, and we will navigate through them. We may not be able to get all of them, but we will definitely try to consolidate and get some questions at the end for all of you. And so please put those in the chat. Thank you.
Awesome. Thank you so much, Marnie. Hopefully, oh, I can share my screen. So let's get this going. Excellent. Yeah, so to introduce myself real quick, so my name is Gemma Carvill. I'm an assistant professor here at Northwestern University in Chicago. And I've been involved with CCC for a couple of years now, from the kind of inception, serving as a role, kind of advisory, scientific role for the last couple of years. And so myself and Heather, who will introduce herself in a moment, are going to tell you a little bit of kind of the state of CHD2 research, and moving forward, the goals of both the researchers, but like how we can partner with CCC and all of you on the call to advance research in CHD2. But before I jump in, I'll just let Heather introduce herself real quick as well.
Thanks, Gemma. Hi, everyone. So as Gemma said, I'm Heather Mefford. I'm so excited to be here and to see all of you who've come today. This is fantastic. I am a medical geneticist by training, both research genetics as well as clinical genetics. I worked in Seattle for a long time, and then about 3 years ago moved to St. Jude Children's Research Hospital, which is in Memphis, Tennessee, for those of you who don't know. So big move, but we're continuing to work on lots of rare genetic epilepsies, including CHD2. And we work very closely with CCC as well as with Dr. Carvill and other people in the field. So we're excited to tell you today just a very brief overview. We're going to give you some big topics to let you know that there are a lot of people around the world working on a lot of different aspects of CHD2. So we're going to tag team this, and I think Gemma's going to kick it off.
Awesome. Thank you, Heather. Yeah, and as Marnie mentioned, we'll kind of go through these slides, and hopefully it'll spur a lot of discussion. And we're happy to answer more questions afterwards. But yeah, the real goal is to give you a big overview of where we are in CHD2 right now. And so a couple of years ago, Heather and myself and a few other scientists worked closely with CCC to think about what is the roadmap to a cure for CHD2. And what are all the things along the way that we need to achieve to be able to realize a therapy, or probably therapies, for this particular CHD2 condition? And so this is put together nicely in this roadmap here. It shows a nice linear progression with a couple of turns in the road. In reality, this is multiple different roads all happening simultaneously. And the way that we've kind of framed the talk today is around these different topics. And so the way we've conceptualized this in a kind of linear manner is that, first of all, the first goal is, of course, to identify who are the patients and families that are affected by CHD2. And so this, of course, is why you are all here today, because this is becoming increasingly successful in terms of both CCC, the Facebook group, genetic testing companies, physicians, genetic counselors, all becoming more aware of the CHD2 condition, and finding ways to bring people together. And so I think this meet and greet today is a great example of how we can bring the CHD2 community together to advance a cure. And then the next thing that we need along the roadmap are really developing models and studying models of CHD2. And so by that, I mean animal and cell models that I'm going to talk about in a little bit more detail in a moment. And then moving on from there, we use these models to then test different therapeutic options in these models and determine whether we think they're likely to work in patients as well. Of course, a key piece of this is not only finding you all out there and bringing the CHD2 community together, but also engaging everybody in research. And so Heather and I feel very strongly about engaging all of you, CHD2 patients, families, in research and telling us what is important to you, but also enrolling in things like studies that help us on the research side to move towards that cure. And so Heather will talk a little bit about involving patients as well and how we're partnering with CCC to try and do that. And then, of course, the ultimate goal is to find cures and new therapies for CHD2. This roadmap also will be published in a manuscript, which will come out in the next year or so, I imagine. And this was really driven by the CCC group in terms of laying out this roadmap. And much of what we'll talk about today will nicely be summarized in this publication that will be coming soon as well. But let's zoom way back to 2013. Yep, a little over 10 years ago, which is strange to think of because it feels like yesterday. So back in 2013 was when Heather and I and a group of wonderful collaborators first identified these de novo mutations in CHD2 and the associated neurological symptoms that you're all intimately familiar with. And so when we first described mutations in this gene, it was really in patients with very severe epilepsies. And so epilepsies with very early onset, very refractory seizures. But we now know that since then that this is definitely not the entire picture of in patients who have mutations in this gene can present with a variety of different symptoms. So different types of seizures, different types of delays, different challenges in behavior. And we know that this condition is much, much broader than what we originally described. And really one of the goals of research in CHD2 is really getting a complete picture of all of the symptoms associated with these CHD2 mutations. And we'll talk about this a little bit more. The other thing that we know about CHD2 mutations is that they, by and large, tend to be de novo. And so what I mean by that is that the variant is not inherited from either parent, but rather it's a new mutation that arises in the child. And so in genetic speak, we say that this is a de novo mutation, so not inherited. And this is one of the hallmarks of most of the mutations in the CHD2 gene. Moving along from kind of identification of the gene to now we've identified the gene. One of the big questions that we have as researchers is when you have a mutation in the CHD2 gene, what happens? What are the downstream consequences of having this mutation? And we know this leads to the clinical, the symptoms in patients. But what we really need are models to help us study the function of this particular protein in the cell. And so we use a variety of different models to do that. We can use animal models. And so a very common model that we use in genetic studies are mouse models, where we can mutate the CHD2 gene in a mouse and then study the mouse to understand the function of this particular protein. And typically, mouse models are the model of choice, but increasingly, researchers now have additional tools in their toolkit that we can use to study this gene. And so there are now also zebrafish models. So zebrafish are very tiny little fish. They're handy to study because they're actually transparent. And so that makes them amenable to a lot of different therapies and studies. And then a newer model is a frog model as well, so where we can mutate the CHD2 gene in a frog model as well. And there are now an increasing number of researchers that are using these models. So, and I've listed them here. I won't go through them each individually, but lots of folks that are increasingly engaged in CHD2 research from the animal model perspective. So animal models traditionally are also really important in terms of testing therapies. But increasingly, we're also starting to turn to cell models. And so this is a human cell model, and essentially where we can do one of two things. We can either take a cell, it's, for instance, a stem cell, which has the potential to become any other cell in the body. So highlighted here would be a stem cell that we can then turn into a neuron, which is one of the major cell types in the brain. Or we can turn these stem cell models into what we call organoids or mini brains. And these are essentially 3D representations of the developing brain that we can then study as well. And so with these stem cell models, we can either mutate the CHD2 gene in the stem cell model, Or alternatively, what we can do is take either a blood sample or a small skin punch biopsy. It's just a tiny little skin punch in individuals who have the CHD2 mutation, and then we can turn them back into stem cells. And from there, we can then turn them into mini brains or neurons. And then we have this model that we can study in the dish and ask questions like, when CHD2 is mutated, how does that impact the protein function, but also how the brain forms and function as well in these organoid models. And so increasingly, these human cell models are being used by a variety of different researchers, including work in my own lab, as well as others, to understand the function of CHD2, but also down the line to test therapies as well. And so this is a really important platform for understanding the biology, but as I said, also starting to do therapeutics in the future, which Heather will touch on. And so with that, I will actually hand it over to Heather.
Thanks, Gemma. That's great. So I'm just going to talk a little bit about some of the more clinical research. And when I say clinical research, I mean, learning from all of you about the challenges that you have with your children, what the phenotypes are and presentation, as well as collecting the samples that we need to do the research that Gemma talked about, like skin or blood for stem cell research. So many of you we've actually touched base with through our St. Jude protocol, where we're enrolling patients. This is called Neurobank. It's just a consent form that allows us to study kids not only with CHD2, but with many other different pediatric onset neurologic conditions. The good news is that now we have an online consent form available that's really easy for everybody to sign. We also have paper consents if that's preferable. And some of you have been meeting with our team to collect some of the data that we're interested in hearing from you about. Do your children have seizures? If so, when did they start? What's worked for treatment? What about behaviors? What about other features that you're concerned about? We also have an option for when we're interested in collecting blood from CHD2 individuals and family members. We can now do that in your home and send someone out to do that. So that's an option. We can also coordinate it with a blood draw if you're getting one for clinical purposes. We've seen a small number of patients here in Memphis we have a neurogenetics clinic that we launched at Le Bonheur Children's Hospital, which is our partner children's hospital here. And we're hoping to kind of take all of the information that we learn from seeing patients and talking to families and comparing that to some of the information that some of you have participated in filling out surveys and providing electronic medical record data through other mechanisms. Next, Gemma. You can do 2, I think. Yeah, so many of you have probably heard of RareX and Citizen. These are 2 different platforms where you can, in RareX, fill out surveys to provide information about your child or your family. And Citizen is an opportunity for you to provide electronic medical records, which actually you just provide the information about where they are, and they will collect those for you. And so we're hoping to kind of coordinate all of this information to get an idea about what we call the natural history of CHD2. And I've just pictured Edith and Emily, who are on my team, who've been doing a lot of speaking with families, consenting and collecting information. And Emily's contact information is at the bottom if you're interested in this aspect of research. Next. Just to let you know that we actually do use this and share it with the greater community. So these are just 2 examples on the left under the red heading there as part of is some data that we presented at the American Society of Human Genetics meeting, which is the largest gathering of human geneticists in the world, really. Happens every year. So we presented some of the CHD2 data that we analyzed from Citizen. And then Stephanie, who introduced herself earlier, very recently presented in the UK at the British Pediatric Neurology Association meeting, data collected from the RareX platform. So we are, as we accumulate this data, we are analyzing it, we're studying it to figure out what we can learn, and also to understand where the gaps are and what we don't know about CHD2 and need to focus on. Next. We're also using those blood samples that you provide for us. So some of you have already provided blood samples through our research program as well as other research programs. And one of the ways that we can use blood samples is to try and understand, is there anything in the blood that we can detect, which we refer to as a biomarker, that might tell us, one, that a child has CHD2, but also can we use that information if we try a new treatment in a child? Does that biomarker change? And can we use that to know if a treatment is working? And this is kind of long-term thinking. But first we have to identify the biomarkers, and then we can figure out if they change. One of those biomarkers is a methylation mark. So this is just it's a mark on the DNA, and it's been shown recently that individuals with CHD2 related disorders have a particular pattern of methylation marks in their blood. And so this is now actually available as a clinical test. So you can actually use it to know whether the CHD2 change that you or your child has is actually disease associated or perhaps not. So this is a clinical test available, and it's been really useful for clinicians and for families. But we're also interested in, CHD2 is not a blood disorder, it's a brain disorder. And so we're interested in understanding, are those methylation marks or that episignature the same in the brain as they are in blood? And while we can't collect brain, like Gemma said, we can use those stem cells to create cells that are brain cells and study the methylation marks in those cells. And that's an active project that actually Gemma and I are collaborating on right now. So that is one of the ways that the blood samples are being used. Some of you have also participated in a study with Combined Brain, which is another group that is focused on lots of rare diseases. And they have been collecting blood as well to look not for methylation marks, but to look more globally at, is there anything in the blood that can tell us this is CHD2, and this is something that we might be able to test to see if it's a marker for response to treatment down the road. Next, Gemma. And then finally, we can use all of these models to try and in all of these models, whether they're animal models or cell models, we have the potential to test new therapies. And the types of therapies that a lot of us in the field are focused on right now are genetic or gene targeting therapies, gene therapies. You may have heard lots of these different terms, precision medicine. And what that means is actually when we know a patient has a CHD2 change causing their disorder, we ask the question, can we go in and either fix that or modulate it in a way that moves it back towards normal? And so the CHD2 gene is in the DNA, which is represented there by the double helix. Yep, thank you. And that star represents a change in the CHD2 gene that is causing developmental epilepsy or other issues. Genes get made into RNA, which is represented on the top there. And remember, everyone has one working copy of CHD2 and one copy that has the genetic change. And so it's the fact that that second copy has a genetic change and we only have half the amount of working CHD2 that is actually causing the problem. So when we think about how do we approach this from a therapy standpoint, there are lots of different creative ways to go about this. None of them are working yet for CHD2, so I'm just going to say that right now. But there are lots of people working on these options and figuring out, do we have something that might work, and can we test it in these models, and can we show that it's safe so that we can then think about moving it into the clinic. One of these is gene editing. So just about everybody now has heard about CRISPR, and CRISPR is a way to go in and actually change the DNA. And so in this case, we would be changing the CHD2 mutation back to the normal copy. This is actually very difficult to do. It works in blood disorders where you can take blood cells out and put them back. It's a little bit harder in the brain when you have to get all of the machinery to make that change into the cells in the brain. Gene replacement is another option. So as I already said, we don't have enough normal CHD2 when you have a mutation. So can we just go in and replace a copy or give individuals who are affected another copy of the gene? So this is kind of the traditional gene therapy or gene replacement therapy that we think about. And this may be an option for CHD2 that people are working on and is available for some other disorders. Other antisense oligonucleotides or ASOs you may have heard about. This is a popular approach that is a little piece of RNA that goes in and attaches to the CHD2 gene and tries to either block you could think about blocking the non-working copy, but that's not enough for CHD2. What you need to do is actually make more of the normal copy. And so ASOs, there are some ways that you can use them to increase the working copy. Another way to increase the working copy is a different type of CRISPR approach, which goes in and sits down on the DNA and turns on CHD2, tells it to make more of the protein. And if you make more of the normal protein, you may actually be able to treat the disease. So all of these types of therapies, and there are others out there, these are just kind of the most popular right now, are actually being tested in different labs across the world to try and help individuals with CHD2. And there, you may have heard, there are some of these therapies available for other rare diseases. Spinal muscular atrophy is one that has both a gene replacement therapy and an ASO therapy. Duchenne muscular dystrophy just had a gene therapy approved, and sickle cell has a CRISPR-based gene therapy that was just approved in the past few months. So as I said, none of these right now are available for CHD2, but they're all in the works and they're all being tested, and we're all trying very hard. And we're just going to end there. These are Gemma's group a couple of years ago with some of the CHD2 board members when the American Epilepsy Society meeting was in Chicago. And this is a photo that we took last year of our group on CHD2 Day in our CCC t-shirts. So I hope that gives you a big picture of what's going on. And some of you have already sent in questions and I see the chat lighting up. So we're happy to spend a few minutes, whatever Marnie, you and Laura think is appropriate, answering questions.
Right. Thank you both. We appreciate that so much. And there were questions that were sent in before. I don't know if Laura, did you consolidate them, or I'm happy just to go ahead and read them if you'd like to do that. Or let's just address the ones that are popped up right now based upon your presentation. And one that came up was, and I'm sure you can read it, but I'll read it for everybody. It says, when you say you can check to see if CHD2 is a disease related, what do you mean? And could you explain it a bit more?
Yeah, so most of the time when you get a genetic test and you get the result back and it says we found something in CHD2, you'll get a report that says this is pathogenic or disease causing. Sometimes people get results back that are less clear. This is true for CHD2 and it's true for other genes where we just don't know enough yet to be able to say we are certain this is causing the things that you're seeing in your child. So that's often referred to as a variant of uncertain significance. And when those, or VUS or VUS, there are lots of ways that people say it, but when you get one of those variants that we're uncertain about, that's when I say we need to do a little more work to figure out if it's actually causing the features or if perhaps it's not, and we need to keep looking for another cause. Does that make sense?
Okay, great. Well, right now I don't see any more in the chat, so I'm going to go to some that were submitted before through their registrations. So let's see. Some of them are, I guess, one of the big overarching questions is, do we foresee a cure? And does the recent research with repairing the gene that causes, like you talked about, sickle cell, give us hope that this CHD2 can be, do we feel like there's a cure on the horizon? I guess they kind of want to hear.
You want to start that one, Gemma?
Sure, I can. If you'd have asked me that question 5 years ago, I would have been very tentative in my answer. But I think that there is reason to be more optimistic today than I would have been 5 years ago. And I think The reason for that answer is because we have so many more tools at our disposal now. So Heather very nicely highlighted all of the different options that we have. Antisense oligonucleotides, gene replacements. And those are all moving at a rapid pace. I think it's very encouraging that other rare diseases are beginning to apply them in clinical trials and in the clinic. But I will temper that enthusiasm with many of those genes were discovered 10 to 20 years ago, even longer, 20, 30 years ago. And there's been a lot of research on these genes, the proteins, and essentially they're a lot further ahead in terms of the research. Some of those proteins are also very well understood, and kind of their normal function in the cell is really well understood. And so that's a challenge with CHD2 in that it probably does a lot of things, and it probably has a different function or role or purpose in different types of cells and at different times during development, during the development of a child from birth to teen years to adulthood. And what we really don't have a good feel for is what exactly CHD2 is doing. And so that's really where on the research side, we're pushing hard to understand what is the quote unquote normal role of CHD2. And also developing all of those models. So that's the other big key piece of this, is that you really need to have a cell or an animal model that you can test all of these therapies in. And so in Science Talk, we call this a readout, and basically a way to test when you then give a drug to a mouse or a cell. Can you then see a reversal of that clinical feature or sign or symptom or phenotype or readout? And so that's where we're really focused right now, in that we don't have those readouts in CHD2. And so I think there is definitely room to be tentatively optimistic, but on the research side, we do have a number of things that we need to overcome first. And the last thing I'll just say, and then I'll let Heather fill in as well, is that in all of those disorders, we know the main cell type that's affected. And often it's something like sickle cell, where Heather said you can take the cells out, repair them and put them back in. But with the brain, it's very, very difficult to do that. And the brain is also a very complex organ. There are lots of different types of what we call neurons and glia, and getting it to the right place at the right time are also things that we need to think about carefully. So with that, I'll let Heather add as well.
Yeah, I agree. I think there are a lot of challenges, but I agree with Gemma that there's a lot of reason for hope. And I think, cure is the holy grail, right? We want a cure. And I think if we think about treatments that are better than they are today, I'm very certain that we're going to keep marching closer to that. And those treatments, even if they're not a cure, will get us closer to a cure. So the other thing to know is that we have a lot more tools at our disposal, like we said, than we did 5 years ago, 10 years ago, including, those gene targeting therapies. Those didn't exist a few years ago. And so now we have these tools that allow us to do things we never thought we'd be able to do. And the other thing I'll say is that I agree with Gemma. Some of these things that are getting cures today, we've known about for 30, 40, 50 years. And CHD2 is in its infancy in comparison. That said, science, the pace of science is also accelerating. And so that is really encouraging. And we just need to be careful that we don't go so fast that we forget to tick all the boxes and make sure that what we come up with is safe for patients. Do you want me to tackle some of these participation questions, Marnie? I think I can.
Sure, I was going to read them out loud, and I don't know if everyone's reading the chat. So the next one, and I think it's important because we have people here from all over the world, a lot of Europe, but we have outside of Europe as well. And one of the key questions is, how can patients give blood, as you talked about? Do you have any?
Yeah. So in theory, absolutely. We can, well, first of all, we can enroll patients from all over the world. That's not a problem to enroll you. To give samples, we just need to figure out how to get that sample, who's going to draw the blood and who's going to pay for that blood draw. And those are things that we can discuss with you. If you enroll in our research study, we can also, if it doesn't look feasible, we can look at whether there are more local researchers who might be studying this. But yeah, we have certainly collected saliva already from patients in Europe. And, if you're seeing a physician regularly and getting blood drawn, we can often piggyback on that, like I said, and we can send you the tubes that you need and the way to ship it back to the United States for us. So that is absolutely possible. The other thing, there was a related question, I think, and I don't remember what it was, but the short answer is yes. It just takes a little bit more logistical machinations.
And that's something I will say, us as an organization, CCC, will help to facilitate that. If, that's our kind of on our goal, our strategic goal is to become more present in the Europe and the rest of the world. And so hopefully we can help accelerate that as well. One of the other questions is, if their therapy were to be developed, would it become available in the UK too, or other places? I think that's a
regulatory question that I honestly don't know the answer to. The short answer is yes, it should be able to be available in the US, and vice versa, right? Things that are developed in Europe eventually make their way to the US. It's a matter of making sure that, again, all the boxes are checked that the government requires to ensure that the drug development jumped through all the right hoops and showed that it's safe for patients. So I don't know how to check all those boxes yet, but those are things that there are lots of people who know how to do that. And once a drug is in clinical trials, then companies often have expertise to kind of ensure that that's going to happen.
I hope you don't mind me mentioning, I know genomics are really keen on ASO therapies and are working on with governments in the UK to try and help. So I think that will pave the way forward for when we will hopefully get one too.
That's great.
I know this is a common question. Have there been any cases proven to be the cause of issues when it's inherited? Is there CHD2, not de novo, I guess?
Do you know the answer to that, Gemma? It
So
is not impossible. It is very rare.
Yeah. And I would say in those instances, that would be a great example where the test that Heather spoke about, the VUS DNA methylation episignature test, would be a great option to determine whether the CHD2 variant is playing a role or not. Yeah.
And just to add that this test is just getting rolled out into Europe. It's only in a few countries right now. Some of them are planning to launch very soon. And so if you have a question on how to get access to that, just send us a message and we'll see how we can help.
Yeah, I just got an email earlier this week even about an inherited CHD2 variant, and my advice was do this methylation test because it will be very helpful in knowing whether this is it or you need to keep looking for a different cause. It's not, no test is perfect, but it's quite a good test and is very helpful in these cases.
Another one is so a lot of people come to the States often, and if they come to visit, can they take part in the testing or studies? And is there a fee? There is
no fee for research. We also don't pay you usually, so it's just the way it works. I think the short answer is yes. And like I said before, even if you're in Europe, there are ways to participate across the pond. But if you come here, we'd love to find a way to see you in person too. Just reach out to us, and one of those I said I saw goes home to Chicago. So Gemma's in Chicago. We're in Memphis. We're easy to find.
Okay. Okay, and then next one, it says, if we manage to get a better version of the gene inside some part of the brain, would we see a difference in our children, or do we absolutely need to bring it to each cell in the brain?
That is the million-dollar question. Million-dollar question. And the short answer is we don't know. The short answer is we don't know. Right now, many groups are working on, so essentially what your question is asking about is delivery, and how do you deliver that gene to as many cells in the brain as possible, and how do you deliver the gene to those regions. And this is a very active area of investigation, not by my lab or CHD2 in general, but there's actually a whole field of study that does just this. And so some of those technical challenges need to be overcome first. And then the second question of how many cells do you need to get into, we just don't know. And that's where having good models, animal models, so the mouse or the zebrafish, but also the cell models as well, is really important. And developing those readouts that will then allow us to tell how many cells we have to get into to kind of change some of those features that we see in the models. So yeah, a very unsatisfactory answer, but where we're at right now.
Okay. The next one just says, is the EpiSign test available in the UK for the VUS? And I guess, was that answered earlier?
Yeah, yes, it is. Yes, it is, and we can get you information on it. I don't even think that there's a web page for it, but we do have the contact info of the person that's coordinating it in the UK.
So Brian, would they get a hold of you through the info at CHD2? Or
Yes.
go through our main info site? And
I'm going to try to look at my email real quick and see if I can get the source in the UK. Give me
okay. And then a UK resident has a daughter and they have a lot of symptoms but no seizures. So how important is it to be seen by medical professionals if there's no seizures? We find no medical professionals know or understand this condition. I think it's
good.
Are you still interested in our kids if they're not having seizures?
Oh, of course. Yeah, absolutely. And that's one of the things that we really want to understand is what we call the spectrum, the phenotypic spectrum, right? We already know that the majority of kids have seizures, but not all. And it's important for us to understand that, and at what ages the seizures develop, so we can give some guidelines to families and physicians. What was I going to say? I mean, understanding that spectrum is really why we want to collect all the information, both from families and from medical records, to know what are the things that your physician needs to know about, right? To be monitoring over the child's lifetime or the adult's lifetime. And I should have mentioned there's a group in Canada who is particularly interested in adults with these conditions, CHD2 and other developmental and epilepsy conditions. So if your loved one is an adult with CHD2, let us know and we can connect you as well. How important is it to be seen? I mean, I think it's important to get regular, just regular pediatric care. We can provide, Gemma and I have helped write a kind of a guide for clinicians about CHD2. There's a website where that's available that just gives general information, kind of what things you need to look for over the lifetime. That can be really useful just to print it out and take it to your pediatrician, especially if you're in a place that really doesn't have a specialty center, so that they have some information and perhaps a way to contact people who might know more. I think, like I said, we're interested in the spectrum, so we want to hear about your kids.
And I will also say that, we at CCC want to have all that information for you as well. So our goal is to also have a lot of that information in a hub for you to print out and take to your doctors, and you can educate your doctors, because we do know that a lot of doctors don't understand CHD2. And then the next question is about, is there a hub being created at Colorado Children's Hospital? And did it happen? There is a chromosome 15 clinic there. In fact, I live in Colorado, so I've been there once a year ago, and I plan on going again this year. And I think they're new and they're up and coming. I can't say they have a lot of data yet, but we have a new Facebook group, and I will share that at my experience there. And I'm happy to share that with all of you. And I'll use this opportunity to plug our new CCC Facebook group that we're launching. So I'll cover that more afterwards, but that will hopefully be a way for us to communicate these hubs as they start to grow, and whether they're worth traveling for. So I will definitely share that.
And I'll
And
just say that I think a lot of people in the community, being kind of the epilepsy genetic neurodevelopmental community, are working to make sure that we have places kind of scattered across the country where there is expertise, so that people can travel a distance, but not too far of a distance, perhaps. We're interested in seeing people, well, anyone, but, you know, the middle of the country is often kind of left behind. And so we're interested in kind of filling that gap. There are great physicians in Chicago. Here in Memphis, we're working on a St. Jude Le Bonheur clinic, you know, Boston, Philadelphia, Denver, San Francisco. There are a lot of opportunities, and yeah, we'd love to work with you too, Marnie, just to make sure that all those are available for patients on the website.
another question is, I'll make sure I don't skip any. Are there any studies looking at boosting the functioning copy of CHD2 as a potential treatment? Do we know of any?
Yes. So that was the slide that Heather nicely outlined, where there's a variety of different ways folks are trying to do that.
Do you have more understanding of why some people with a mutation in CHD2 do not develop any neuropathology?
I think if there's zero neuropathology, I would make sure that CHD2 is the cause. And if you're talking about a parent, for example, this is another example where I would test that using the methylation test just to make sure, because you want to have the right diagnosis, whether it's CHD2 or something else, right? But you know, more broadly, we don't understand why some people have seizures and some people don't, why some people have severe behavioral challenges and some people don't. So those are all things that we're trying to understand. That's why we're collecting all the clinical information. Is it related to the mutation? Is it related to something else in the DNA? That we just don't know the answer to that. And it's true for CHD2, but it's also true for many other genetic conditions, that there's a wide spectrum, and it's a lot of work to figure out why.
And this was asked earlier, and it says, can you use stem cells for studies instead of blood draws? A lot of people have stem cells of their children, and so can they be used?
Do you know, Gemma?
In theory,
Yeah.
in theory, you can reprogram almost any cell back to a stem cell. So I think in theory they can be used. Yeah,
We haven't actually
we personally
tried.
have not done.
Yeah.
Yeah, we personally have not done it. And I don't know anybody in this kind of rare disease space that has, but in theory, I think it would be possible.
A lot of them are not really. This next one's not really. I don't think something you can, but I'll just put it out there. If a cure is discovered in the future, could we anticipate improvements not just in seizures but behavioral and also intellectual developmental disabilities? I would say
That would be the definition of a cure right there. Yeah, no, I think that's the hope, is that, you know, oftentimes we focus on seizures because that's something that we can quantify a little better than some other things. But the hope is that if we can actually get at the root cause of the condition, being the CHD2 gene, that whatever treatment we have would help all of those aspects. Now, it might not be perfect depending on how old you are when you get treated, right? But I would expect that what we hope for is that there will be improvement across all of those aspects.
I also let everybody know there's some links in the chat if you'd like to link to what Heather and Laura have provided. So get those. And then, of course, people are looking for doctors, and that's Landis in Europe and Australia says, are there any doctors here we can work with that can provide you with information and test results? I guess it's, you know, are you guys?
Yeah, we can work with anybody, but specifically in Australia, there are some really good epilepsy genetics folks, geneticists. So depending on where you are, we can perhaps direct you to the right clinics. But we're happy to work with them. And actually in Australia, you should know there is, I think there's a big natural history study for genetic epilepsies that's led by Dr. Sheffer. And so CHD2 is one of those genes that they're interested in. And so that may be an option as well. And we work closely with her also.
Okay, there's one written in Italian that I'm going to have to translate.
She was also asking about whether there was something in Italy.
There are some good epilepsy folks or genetics neurodevelopmental folks in Italy too. We'll have to, maybe we can try to come up with a list for you.
Yeah, I've started collecting a list. We'll have to work together and
Yeah,
and then make it public.
yeah.
Yeah, post it somewhere we can add. Yeah.
And this is a good question because we have a lot older, more older people being diagnosed. So broadly speaking, the older you are, is it harder to apply gene therapies in conditions like this?
We don't know the answer to that, to be perfectly honest. And that is, that's a question that we'll answer over time. I think there's no reason not to think. There have been some studies in other conditions where adults are diagnosed late in life and they're treating the seizures appropriately, for example, and there's improvement in, you know, cognition and kind of interaction. It's not a cure, but that's a different type of therapy. But I think there's reason to believe that there would be improvement, but we don't know the answer.
And I have a last question here. What are the factors that contribute to the speed with which the research develops? Are there things other than signing up for studies that we can do to support you? Great question.
Excellent question. Hard question.
Take it away.
The factors that contribute to the speed with which research goes, I worked in this space for a while now and have grown to appreciate that it's hard to explain how long research really takes. And so I think, like, to provide some context, I spoke about those little mini brains that we make. And so those, ideally, we're growing those in a dish for 6-7 months sometimes even longer, up to a year. And that's just growing them in the dish. That's without doing any sort of experiments, anything essentially. So you're waiting a year before you can even do an experiment, and that's really frustrating for patients and families. It's super frustrating for us. If we could make it go faster, we would. So I think some of what seems to take a really long time in research, and that we're not moving fast enough, is honestly just often a technical limitation of what we're trying to do. It just does take time. In terms of what can we do to speed things up, I think the types of things that you all are already doing, being engaged in clinical research, being engaged in Heather's DNA methylation study, for instance. Honestly, other than time and money, the only other big thing that we need is lots of data in terms of clinical presentation and in terms of samples, knowing where everybody's genetic mutations lie within the gene, because that might tell us things like if patients have mutations that tend to cluster in this part of the gene, maybe they have a more mild phenotype or less clinical features. And that helps us because that tells us something about the protein structure. And so, again, another very unsatisfactory answer, but honestly, that's one of the biggest things I think that you all as a community can do. So yeah, that's how I view that question.
And I would just, some people don't like to talk about it, but I would emphasize money. I think that funding research takes time. It also takes people, and people are expensive. And that's just the truth of it. And that's something that I've come to learn over time as well. And the CCC has done a great job of fundraising, and I really applaud your efforts. And that goes often to funding young investigators, which is important, because getting young, excited people with new ideas engaged and committed to CHD2 means that they might be the ones who have the next big discovery, and they might study it for their career, right? So that's important. And I know, like, there was fundraising, I think, around your this event today, and I think that's great. So it's not all money, and I think the things that Gemma mentioned are as much, if not more important. And we'd love to have you all engaged in research. It is highly important, right, to know that. And yes, Dr. Guarini is an expert. He's wonderful. So it's in the chat. So I think, all of those things, time, money, contributions of samples and of data are really important. And it does get faster, and we learn from other diseases. So both Gemma and I work on other things and talk to other groups and rare diseases, and we learn from each other, which I think is important as well, because maybe what we discover as a potential approach to a cure for CHD2 might work for 10 other disorders. Wouldn't that be great, right?
And I'll say that plugging on with Heather, she, last year we funded, and I saw on your slides, Heather Wilsey's frog model, and I didn't see, I may have been not paying attention to it, and we had 2 early investigator grants that we funded last year. So I just want to let you know that's where the money that you guys are raising is going. And then we're also in the middle of writing grants in order to get more money to give to researchers. So I just want to tag along with what Heather said. And then let's see. I feel like there's a conversation going on in chat.
Yeah, there's another question about, now that we have more patients registered, are there studies about which AEDs work, I assume specifically with CHD2 as opposed to more generally?
Yeah, we're collecting that data. It's really complicated and it's not clear, to be perfectly honest. And a lot of people get treated with different medications at different times and different combinations. And that's one of the hardest things to kind of tease apart. But that's why we collect the data, as we're trying to figure that out.
And I'm going to put in a plug. That's why we asked you to fill out RareX, and why we asked for your citizen data. And we're going to talk a little bit more about that, but that is why we really need everybody to share their data, because that's how we're going to figure it out.
There's a really good platform called Citizen that you've heard mentioned a few times. And what Citizen does is, instead of you filling out a bunch of surveys, it just pulls your entire medical record. The problem is it's really only available in the United States right now. And the biggest reason is that there's a bunch of the laws in the United States sort of support you getting your data easily. Other countries aren't so easy. And so we think it's going to be rolled out in more countries. It's just taking a long time, and that could really accelerate. And so anytime we have a research opportunity, we're looking to see where we can apply this around the world. But unfortunately, with various things, there's certain roadblocks, and we just have to deal with them.
Right, and that's so there's one more question. It's more, I think, our seizures are only early onset, or we're told to wait to watch increase in seizures do occur. Are there any data on seizures starting later in life? I think that is already can be answered with yes. They start early, some start later, but it seems like a majority of them start early.
I would say the majority start early, but there have certainly been kids who have seizures start later, 6 years, 8 years, even 12 years. So that's less common, but it does happen. And I'm going to have to jump off. I'm sorry, but this has been fantastic.
About an hour. And so I'm going to let Dr. Carvill and Dr. Mefford go. And I really want to say thank you for your time today. And I hope everyone else stays on for a few more minutes to just have some great some information I want to share with the rest of you. But we'll try and do this again. And just so everyone knows, we stay in touch with Dr. Carvill and Dr. Mefford all the time. We're constantly talking to them about research studies, funding, grant proposals, all kinds of stuff. So just know that that's what we're doing at CCC, is trying to bring all of our communities together, and so we can all connect and learn and move the research forward. So we really appreciate your time.